Hey students, it's Mr. Luchero. I just wanted to create this recording again of the lesson we did this morning, Unit 3, Lesson 2. So again, there were two objectives to this lesson. The first was just to review enzymes and relate them to energetics, which we talked about in lesson number 1. And then the second thing is I talked about a couple of different factors that can impact enzyme function and therefore the rate of reaction that the enzyme catalyzes. So let's start off with that first objective here, right? So first, what is an enzyme? So an enzyme is a protein. It's extremely important that you remember that an enzyme, the type of molecule it is a protein, and its function is to catalyze a reaction. And what that means is that an enzyme speeds up the rate of reaction by lowering something that we call the activation energy. Or E subscript A for short. Okay, now it's important to note that catalysts is another term that we'll see. It's the same thing as saying an enzyme. Okay, so if we take a look at this diagram right here, we can unpack a little bit about this definition. So this is a free energy diagram. We've seen lots of these in lesson number 1. In class, I asked if we could identify if this was an endergonic or an exergonic reaction. And based on the observation that the reactants have more free energy than the products, therefore delta G is less than 0, we can say that this is an exergonic reaction. Okay, now remember that the role of an enzyme is that it is a protein that catalyzes the reaction by lowering activation energy. So just a quick recap. So activation energy, or E subscript A, is this barrier that the reactants have to get over before that reaction will proceed. Energy has to be absorbed from the surroundings in order to get those reactants above that activation energy hump, and then it will proceed downhill from there. What a catalyst or an enzyme does is it will lower that activation energy from originally being represented with this purple line to where it is with the green line. And so that actually allows the reaction to happen a lot quicker because the barrier is a lot lower. And here we see, right, these labels, the catalyzed reaction, that's referring to when the enzyme is present. Okay, so we've got relation to our energetics, or our free energy diagrams. Let's talk about the types of enzymes. So there's two major types of enzymes we're going to see. There are those that are involved in catabolic pathways, which again are the breaking down of molecules. These are typically exergonic in nature, and so we can have enzymes that are catabolic. And then the opposite, if you remember, would be something like anabolic pathways. So anabolic enzymes, they help to, instead of break down molecules, they help build larger molecules. One of the big examples of an anabolic enzyme in biology is something like DNA polymerase. DNA polymerase is used to build a DNA molecule during DNA replication. It builds a larger molecule, and if you remember, anabolic reactions, they tend to be endergonic in nature, and catabolic reactions, they tend to be exergonic. An example of a catabolic reaction, which we'll see in this unit, is something like catalase. It breaks down hydrogen peroxide into smaller parts, into water and oxygen. Okay, so those are our types. Let's talk about structure. So structure, there are two major terms when we talk about an enzyme. The first is that on the actual enzyme, we have something called an active site. It's a physical location on the enzyme into which the substrate fits perfectly into. So that substrate is typically a reactant, all right, from the reaction that this enzyme is catalyzing. And when that substrate binds, the reaction can occur with a lower amount of activation energy. Okay, so that's the major structural component. We have active site and it binds to a substrate. Now remember that an enzyme is a protein. And if you remember from Unit 1, the enzyme structure is determined by the order of amino acids which influence how the protein folds. Right? And we know that the order of amino acids, that's ultimately determined by the DNA sequence. All right, through transcription and translation, ultimately the information that was needed to make a protein and determine the amino acids of the protein came from the DNA. Okay, so this enzyme structure, again, consists of an active site. The way that the enzyme folds is dictated by the amino acids, which will give that active site a very specific site for a substrate to bind to. Okay, so let's go on to our second objective here. Okay, so let's talk about the second objective. So the second thing I talked about was different factors that can impact an enzyme's function and ultimately the rate of the reaction that it catalyzes. So in the video lesson that you saw from the boson video, you learned about two different types of things that can inhibit enzyme function. And we'll start there. So in this figure right over here, we have an enzyme that is binding a specific substrate at the active site. Now there are molecules that can actually prevent that substrate from binding and ultimately prevent the enzyme from doing its job or inhibit the enzyme. And the first category of molecule that does that is something called a competitive inhibitor. And competitive inhibition is where that molecule, represented in blue here, has a similar shape in some ways to the substrate that allows it to literally compete for the active site. It'll bind to the active site in place of the actual substrate, which prevents the substrate from binding and ultimately the enzyme doing its function. The other category of molecule that accomplishes the same thing is something called a non-competitive inhibitor. And while the result is the same, the mechanism is different. In this case, the inhibitor molecule doesn't bind to the active site, which again is up here, but instead binds to what we call an allosteric site. This is also called allosteric inhibition. It's just a site, a location on the enzyme that's different from the active site. And when that binds, it actually causes a conformational, a structural change to the enzyme that makes the active site no longer able to bind the substrate. And so, again, the result is that the enzyme can't function. Now, something that I mentioned as well is there's something called feedback inhibition that's very heavily tied to especially non-competitive inhibition. And the idea is that if an enzyme is working in a cell to allow a particular reaction to proceed at a faster rate, the cell might not need that reaction to go on continuously, right? It might only need it at a certain time, and it might be wasteful or even harmful for that reaction to continue indefinitely. And so feedback inhibition is actually where the product or one of the products of the reaction being catalyzed acts as a non-competitive inhibitor. Okay, and if you think about this, it makes sense, right? If you have an enzyme that's catalyzing a reaction, right, turning the substrate into the products, right, when that reaction has occurred sufficiently and there's a good amount of the product, it would make sense that one of the products of the reaction could serve as a non-competitive inhibitor to slow down the activity of the enzymes, right, by binding to that allosteric site and preventing that reaction from continuing. And when you run out of products, you no longer have these non-competitive inhibitors, and so then the reaction picks up again. So that's what we mean by feedback inhibition, is where the products of the reaction literally provide feedback to the enzyme to regulate it or slow it down when necessary. Okay, so that's the first thing, is this idea of inhibition, competitive inhibition, non-competitive inhibition, and feedback inhibition. Something else that can affect enzyme function is a mutation. So if you remember that an enzyme, like we see right here, is a protein, and a protein folds in a particular manner. In this protein, we see some alpha helices, some beta pleated sheets, and all of that is dictated by the order of amino acids, all right, which I mentioned previously is again determined by the DNA sequence. And so a mutation is a change in the DNA sequence. And because that controls the amino acids, by changing the DNA, you can change the order of amino acids and ultimately change the structure of the enzyme. And since we know that an enzyme relies on its active site in order to function normally, this can ultimately affect its function if its shape or structure is affected. Okay, so mutations can harm enzyme function. All right, now the other thing that can affect enzyme function is something called denaturing. And denature, if you look at this figure right here, is summed up as meaning that the enzyme, when exposed to different conditions, will begin to lose its proper shape. And so we see a normal enzyme and a denatured enzyme, and there are a couple of things that can cause this to happen. I'm going to go over just a couple of them. So the first thing that can cause an enzyme to denature or lose its shape is a change in temperature. Especially if you increase temperatures, what that can do is it can disrupt the bonds and the interactions between amino acids that make up the enzyme and can cause the enzyme to lose its shape and ultimately denature and no longer be able to function. Most enzymes in our body, they operate at an optimal temperature that matches our body temperature. And so if you go above that significantly, and if you go below that, it can disrupt those bonds and cause the enzyme to denature. Okay, the second thing that can cause an enzyme to denature is a change in pH. And if you remember, right, pH is a scale from 1 to 14, with 1 referring to conditions that are acidic or very acidic, and 14 referring to an environment that's very basic. Now, most enzymes in our body, our body holds a pH of around between 6 to 8, with some exceptions depending on the part of the body we're talking about. And so if you go above that significantly or beneath that significantly, it can cause, in a similar way to what temperature does, it can disrupt the interactions within the protein and cause it to denature. So we'd say a change in pH can cause an enzyme to denature. Now, the third thing that I mentioned that's pretty rare, but just want to mention here, is a change in the salinity of the environment, meaning how much salt is present. And if you remember how salt, the structure of a salt molecule like NaCl or potassium chloride, they're composed of ions. And when they dissociate, those ions hold charges, which again can disrupt in some ways the interactions in an enzyme. Okay. These first two we're going to see a lot more often in this unit, but salinity can also cause an enzyme to denature. All right, now all three of these examples, right, they've all negatively impacted enzyme function and ultimately slowed down, if not completely stopped, the rate of reaction that the enzyme is catalyzing. I want to talk a little bit about something that can actually increase the rate of reaction. So one of the things that can increase the rate of reaction is if you increase the amount of substrate. So remember, substrate is what attaches to the active site. And in this diagram right here, we have enzymes, which are these red structures, and substrate, which are the blue structures. Now, if you increase the amount of substrate, it makes sense that you're going to increase the rate of the reaction that's happening, right? Because there's more substrate that can be attaching to these active sites and going through the reaction, and so you can expect a higher rate of reaction. Now, this only occurs to an extent, though, and to kind of help explain that, let's take a look at this graph here. So we can actually measure the rate of a reaction, an enzyme catalyzed reaction, in two ways. The first way we can measure the rate of a reaction is how quickly the substrate is being used up, or the reactants are being used up. The other way we can measure it is how quickly products are being formed, right? That is after the substrate has been attached to the active site and the reaction has occurred. So those are two different categories of way we can measure the rate of reaction. In this graph, it doesn't specify, but either way, right, a faster rate of reaction is associated with faster use of substrate and faster creation of product. On the x-axis, we have substrate concentration, which again is referring to, kind of in this model, increasing the amount of available substrate in the environment. And as you notice, right, the rate of reaction increases as substrate concentration increases, but it only increases to an extent. It actually plateaus off. And this is actually what we refer to as the Vmax of this reaction. And this is simply where this is the maximum rate of reaction when all active sites are saturated, right? Because if you think about this figure right here, right, if you only have a certain number of enzymes, but you have an infinite amount of substrate, there's going to come a point where all of the active sites are occupied. And if the amount of enzymes stays constant, there's a max rate of reaction that can occur, because there's just simply going to be a saturation of the active sites, and then as soon as those active sites are open again, will be replaced by new substrate. So there's a max rate of reaction at that point. Now you can increase the Vmax of a reaction, but that requires often increasing the amount of enzymes in order to do that. Okay, so the amount of substrate can impact the rate of reaction, as well as the amount of enzyme can impact the rate of reaction. So again, those are the main things that I talked about in today's lesson. I hope that was helpful. Please reach out if you have any questions or need me to clarify anything for you. And I will see you on Friday.